Welcome, guys. Today we are doing the topic on developing drugs. So I have prepared this presentation for you, and we're going to focus on what are the key words and key points for your exam and mark scheme. So before we start, developing drugs, what does that mean? So basically, what it is, you have a new drug, like a new medicine, it could be a new vaccine. That's drugs. Now, before these medications, they are in the shops, they are in the pharmacy. Before people begin to use them, they need to go through different stages, and this is what we're doing today. What are the stages that a drug needs to be going through? So let's start. We have our over here, let me move my picture. We have two stages. The first stage is a preclinical stage, and the second stage for exam is called the clinical stage or the clinical trial stage. So what do we need to know for the preclinical stage? For the preclinical stage, it happens in a lab, in a laboratory. That's very important. You have to mention the word. It happens in a laboratory, the preclinical stage. So this drug, this new medication, is tested on these three things: cells, tissues, and live animals. Stating these names gives you another second mark. Happy with that? Now, why are we testing on these three things? By the way, for animals, you have to say live animals. If you say animals, you don't get a mark. The animal has to be alive. Now, we are testing on these three things in a lab to test for toxicity, efficacy, and dosage. These three things, mentioning all three of them gives you another 1 mark. Now, you can have a separate question: what is toxicity, what is efficacy, and dosage? Let's quickly go through that. Now, toxicity means how harmful this new medication, this new drug is, and does it have any side effect. So toxicity means how harmful it is. Efficacy, it's not efficiency, efficacy. Efficacy means whether this drug actually works, whether this drug can cure that disease. As efficacy, does it even work? And we are doing that test in the preclinical phase. And the third thing is dosage. Dosage is what concentration of drug is needed for this medication to work. So these are the three things we are testing. Now, once we are happy with this, once it has passed that stage, what stage? Preclinical stage. Then we go into the clinical trial stage. And in this trial, it's called clinical. It involves human beings. You should mention healthy volunteers and patient. That gives you a mark. Healthy volunteers and patient. Now, there are 2 steps, and you need to mention that in order. In this phase, in this stage, in the clinical trial stage, these healthy volunteers and patient, we give them a very low dose, small amount of this medication. Why are we doing that? To monitor for safety, to see side effects. That's another mark. If we are happy with the side effect, meaning that it's not going to kill that patient or person, the side effect is not really bad, might be like a fever, we can go into the second part, is do further trials, do more trials on patients. That's another mark, further trials. Why are we doing further trials? We are doing further trials, testing this medication on more people to find optimum dosage. That's what we are doing. We are trying to find optimum dosage. Now, what is that? Optimum dosage, this is the dose of drug. So this is the amount of drug that is need to be given, but is the most effective. How much amount of drug should the person take in order for this medication to be the most effective? That's optimum dosage. And also we are testing for efficacy. That's another point. And efficacy means that is this medication going to work and cure or treat this disease or illness? Now, whilst you're doing the clinical trials, further marks are here in these. Can you see these things? Let's go through them. You're going to make sure that this is a double-blind investigation or double-blind trial. So what does that mean? That means you're going to have two groups, group 1, group 2. And you have to mention what each group is to get the mark. Group 1 will have this new medication. And group 2 will not have the new medication, will have a placebo, like a fake drug. But why is it called double blind? It's because group 1 and group 2, they don't know if they are receiving the actual drug or the fake one, the placebo one. That's why it's called double blind. Now, randomly, what does that mean? We're going to, who will decide which patient goes to which group? Does the patient go to the placebo group, fake drug, or the actual one, actual drug group? It's random. We randomly allocate patients to which group they should go into. After the end of all of our investigation and clinical trials, we have all our data. We're going to make sure our data is peer reviewed, meaning someone else, other scientists are going to come and they're going to check our results before they are published. Why do we do that? Is to prevent biasness and prevent false claim. So this is all you need to know, and it's really exam focus.